Did you know that quantum biology has been around for almost 100 years? Could the rules that govern subatomic particles actually help explain how living systems work? Today, researchers are investigating whether quantum phenomena play a role in everyday biological processes, like how plants harvest sunlight through photosynthesis, how enzymes speed up chemical reactions, how migrating birds navigate across continents, and even how mutations occur in our DNA. But these ideas didn't appear overnight. The history of quantum biology stretches back nearly a century. It began with physicists asking broad philosophical questions, moving through decades of skepticism, and eventually grew into an experimental science being tested in actual modern laboratories. So how did quantum biology begin, and how did it develop into the field that we know today? Let's take a look. At the beginning of the 20th century, physicists discovered that the microscopic world is not governed by the Newtonian physics that describes our everyday experience, but by a different set of rules described by quantum mechanics. Energy came in discrete packets called quanta, and subatomic particles could exist in states of probability, behaving both like waves and particles. By the late 1920s, quantum mechanics had matured enough to describe atoms, electrons, and chemical bonds. After transforming their understanding of non-living matter, some researchers began asking, could these same principles reveal something fundamental about life itself? One of the earliest connections between quantum physics and biology came in 1929. Physicist Niels Bohr discussed what the new understanding of atomic phenomena might contribute to the study of living organisms. Bohr developed these ideas further in his famous 1932 lecture, Light and Life. He didn't propose a specific quantum mechanism inside of living cells. Instead, he explored whether concepts emerging from quantum physics might also shape how scientists understood the complexity and organization of living systems. Around that same time, German physicist Pascual Jordan began developing more concrete ideas about how quantum physics might interact with life. And in 1932, he published what is often considered the first scientific paper on quantum biology. Jordan argued that quantum events affecting a small number of biologically important molecules might be amplified until they influence an entire organism. And a quick historical side note here, Jordan's work carries a complicated legacy. He was an active member of the Nazi Party and later tried to align his biological theories with Nazi ideology. So Jordan's support for this did deeply damage his legacy. After the war, his work largely disappeared from mainstream science, while rises in molecular biology also helped push broader quantum biological speculation into the background. Despite this setback, arguably the most influential moment arrived in 1944, when the famous physicist Erwin Schrödinger published a book titled What is Life? Schrödinger argued that classical physics wasn't enough to explain how traits were passed down through generations with such incredible precision. He proposed that genetic information must be stored inside a tiny structure governed by quantum stability, which he called an aperiodic crystal. He even suggested that random genetic mutations might be caused by quantum particles jumping out of place. Schrödinger's book helped inspire an entire generation of scientists to help bridge the gap between biology, chemistry, and physics. In 1953, James Swanson and Francis Crick, influenced by Schrödinger's idea, proposed the double helical model of DNA, and biology changed forever. The structure immediately suggested how genetic information might be copied, while discoveries over the following years revealed how cells replicate DNA, read genetic information, and produce proteins. The rapid rise of molecular biology meant that life could largely be explained through traditional biological structures and chemical reactions. For a long time, speculative quantum ideas were pushed to the background. Standard biology was moving fast enough without needing extra help from physics. Yet, quantum physics didn't disappear completely. In 1963, Swedish physicist Per-Olof Lodin revived Schrödinger's mutation idea. He proposed that protons inside DNA might be using quantum tunneling. In classical physics, a particle needs a certain amount of energy to push over a barrier, like a ball rolling over a hill. But in quantum physics, a particle may occasionally tunnel right through a barrier, even if it doesn't have enough energy to go over it. Lodin argued that if a proton tunneled across a DNA base pair right as the cell was copying its code, it could trigger a genetic mutation. While scientists are still debating how often this actually happens inside cells today, this provided one of the field's first specific and potentially testable molecular mechanisms. By the late 1960s, experimental evidence for specific quantum effects in biological systems was beginning to emerge. The first major clue arrived in 1966 when researchers Don DeVault and Britton Chance studied electron transfer in photosynthetic bacteria. They found that at low temperatures, the electron transfer rate became almost independent of temperature. That behavior was difficult to explain through ordinary thermally activated barrier crossing, and provided strong evidence that the electron was tunneling through the barrier, and a first experimental hint at tunneling in a living system. Around that same time, physicist Herbert Fröhlich provided another possibility. He suggested that energy supplied by metabolism could become concentrated in a shared low-frequency vibrational mode, producing long-range coherence in biological molecules at body temperature. And another important development came from spin chemistry. In 1969, researchers developed the radical pair mechanism to explain unusual signals observed during chemical reactions. Their work showed how the evolution of electron spins could influence how chemical products formed. Building on this foundation, physicists Klaus Scholten, Carl Schwenberg, and Albert Weller proposed a biological application in 1978. They suggested that radical pair reactions could be altered by the direction of Earth's weak magnetic field. So if an organism could detect the resulting chemical changes, it may provide a directional signal. And this proposal became the foundation of the modern radical pair hypothesis for magnetoreception in animals. But a lingering objection remained, that living cells are too warm and chaotic for fragile quantum states to survive. But by the late 1980s, researchers were establishing that quantum tunneling could contribute to enzyme-catalyzed reactions even at room temperature. In 1989, Judith Klinman's group reported strong kinetic evidence for hydrogen tunneling in an enzyme reaction, helping establish tunneling as a measurable component of some biological chemistry. By 1996, biophysicist Luca Turin pushed the boundary even further by proposing a quantum mechanism being involved with smell. He suggested that our nose receptors identify scent molecules not just by their physical shape, but by their molecular vibrations using electron tunneling. It's still a debated idea among olfaction researchers, but it opened up sensory biology as another place to look for quantum effects. By the 2000s, quantum biology was finally receiving renewed attention from researchers across biology, chemistry, and physics. In 2000, Thorsten Ritz built upon earlier bird navigation theories by pinpointing a specific light-sensitive protein in avian retinas called cryptochrome as the actual engine behind the radical pair compass. Today, this remains the leading scientific explanation for how migratory birds move across the globe. Then came a major breakthrough in photosynthesis in 2007. A team led by Gregory Engel and Graham Fleming observed evidence of long-lived quantum coherence in a light-harvesting complex from green sulfur bacteria. Their results suggested that energy could move through the complex with wave-like quantum behavior, exploring multiple transfer pathways as it traveled toward the reaction center, where the energy can ultimately be converted into chemical energy. And this discovery helped kick off global research. In 2009, the first Quantum Effects in Biological Systems workshop, also known as QUEBS, was held in Lisbon. It's become a recurring international meeting for researchers across physics, chemistry, biology, and related fields. By 2014, scientists Al-Khalili and Johnjoe McFadden published Life on the Edge, a book synthesizing decades of quantum biology for a mainstream audience. Dedicated training programs, research centers, and conferences have since helped turn quantum biology from a scattered collection of questions into a more connected, interdisciplinary field. So where does that leave us today? Over the past decade, quantum biology has moved from a fringe scientific curiosity to a rapidly expanding frontier. Researchers are using modern quantum sensors and advanced laser spectroscopy to look deeper into living systems than we ever could. If we can fully understand how nature utilizes room temperature quantum effects without needing expensive deep freeze conditions, it could reshape parts of our world. It could point toward more efficient solar energy capture inspired by photosynthesis, new directions in drug discovery targeting quantum interactions in proteins, and new kinds of bio-inspired sensory technology. Almost 100 years after Niels Bohr stood in a lecture hall asking, could quantum physics apply to life, we're finally building the tools to test those questions and find the answers. If you want to follow along, support cutting-edge research, or join our community, make sure to check out the description below and hop into our Discord to check out the Quantum Biology DAO. Give us a subscribe if you want to keep following along, and may the quantum be with you.